Today on iConnect, I'm joined by Professor Sarah Coupland, consultant histopathologist and president-elect of the Royal College of Pathologists. Sarah is one of the world's leading experts in ocular pathology, and her work has helped us understand what our tumours can tell us about ocular melanoma, from genetics and predicting risk to why some cancers behave differently from others. Today, we're going behind the scenes to explore what actually happens to our tumour samples, the extraordinary value of the Liverpool Ocular Oncology Biobank, and how the tissue patients donate today could help shape the treatments of tomorrow. Sarah, thank you so much for joining me today. I can see you're at the hospital, are you?
Yes, I am. Yes, in my office. So I've got the NHS side over there and the university side here.
Fabulous. So before we get into the science, the hospital, the medicinal, the pathological side of this, Sarah Coupland, Professor Sarah Coupland, where did you start and what on earth got you involved in ophthalmology, especially ocular oncology?
Oh, thank you very much, Joanne, for inviting me to this podcast interview. It's a real pleasure. About myself, so I originally come from Australia, and I did my medicine training in Sydney. And then I thought that I might do ophthalmology. And so I did my junior doctor years for 2 years and, did a variety of different subspecialties. And then I met my future husband, who's German, and I then moved to Berlin and did a PhD while I learnt how to speak German. And this PhD was essentially in ophthalmology. I did a variety of different things, but during that PhD, I decided that actually what intrigued me most was the underlying biology and mechanisms of disease. And so I decided that I'd actually do pathology training. And so I did my pathology training in Berlin, 6 or 7 years in length. Of course, there was a baby break in between. And yes, I always had an interest in ophthalmic pathology as a result of my PhD. And where I did my training was a referral centre for eye cancers. And not only did I do my general pathology training, but I also continued separately doing ophthalmic pathology, and so saw retinoblastomas, conjunctival melanomas, and of course uveal melanomas. And then we moved here to the UK, and that's nearly 21 years ago now. And there was a specific post for an ophthalmic pathologist, which is a bit unusual, but because ophthalmic pathology is a rare subspecialty, the NHS has highly specialised services, sponsors for ophthalmic pathology posts in England. And I was lucky enough to get one of those. But right from the very beginning, I was able to organise a job plan, which was a mixture of both research and NHS diagnostics. So I came in as a senior lecturer, and my task at that stage in 2005 was to set up my own research group and bring in grant money and do research. And at that stage, when my husband and I both moved here, it was Professor D'Amato who was running the Ocular Oncology Centre. And Prof. D'Amato, or Bertel, he had tremendous foresight, and he started building a database, a clinical database in Liverpool. And when it came to research, we just clicked immediately. We both wanted to understand better what was the underlying biology in eye cancers, but particularly in ocular melanomas. And so we together formed what continues to be called the Liverpool Ocular Oncology Research Group. And yes, we'll go on to further things, but one thing led to the other, setting up the Ocular Oncology Biobank and organising positions for postdoctoral scientists. Dr. Helen Callery, who's been with me for many, many years, and a whole series of PhD students who all have different specific projects and, looking at various aspects of eye cancers in general. So I know that was a long spiel, but essentially, yes, I guess I moved at the right time into the right environment, and with the tremendous support of Prof. D'Amato and others, I was able to write lots of grants and bring in an income to set up this research team. And simultaneously, on the NHS side, together with the two ophthalmic pathologists who were there at that time, I was able to really do deep dives into ophthalmic pathology. But I should just mention that I did do other diagnostic reporting. So another great love of mine is hematopathology. So that's lymphomas and leukemias. And so I have actually, when I was in Berlin, I wrote what would be considered like an MD, sort of like a doctoral thesis, all about ocular lymphomas. So not melanomas, ocular lymphomas.
I have never heard of an ocular lymphoma.
Yeah. So you can, yes, you can get lymphomas in and around the eye. And the ones around the eyes tend to be less aggressive than the ones that occur inside the eye. And the ones inside the eye are very tricky to diagnose, and unfortunately are usually associated with brain lymphomas. And so very tricky to treat.
No interest to getting in there with a scalpel and getting in amongst it while it's in the human.
So, I mean, I did, when I did my PhD, I did do corneal transplants, and I really, I think I'm quite dexterous, but I just decided that I really enjoyed understanding the nitty-gritty of what was going on behind things. And I guess it probably comes from the family history type side of things, in that my father was a medical oncologist. And I sort of
grew up. That explains a lot, Sarah.
Yeah, so I grew up, so my father was medical oncologist, my mother a nurse. And so you can imagine the conversation around the dinner table, when I was growing up. It was very medical. And so, yeah, I think I probably had more of an interest in the theory and the biology of how to treat disease rather than a surgical. I mean, I had a surgical interest, but I think I preferred the medical and the biological sort of side of things.
Well, we're very grateful to have you in that position. I mean, most patients know that a tumour sample kind of disappears off to a laboratory. You can sign it away at the point of either having your eye removed, or if you're offered biopsy, when you have your ruthenium plaque. What actually happens to it? I mean, literally, does it land in an in tray in your laboratory? And if it does, then where does it go?
So I think when people hear about this specimen being sent away, it's sort of almost like it's landing in a black box, and then a result comes out, however many days later. And sometimes the language used is a wee bit difficult to understand, and pathologists perhaps are a wee bit pedantic with their terminologies and their labeling of things. So what happens usually, let's speak about perhaps an intraocular biopsy, for whether it's a diagnostic one or a prognostic one. Typically that biopsy is taken by the surgeon, and then it goes into a transport container, like a little container, which is labeled up with the patient's name. And it's usually, the biopsy is put in a transport medium. So it's a liquid which it either preserves it straight away, so something called formalin, or it can be sent in just a transport medium so that it then arrives in the laboratory ready to be worked up straight away. And accompanying that pot or container is a request form. with the patient details, usually a bit about on the form, what the clinical history and what the clinical question is. Okay.
Oh, give us an example of those questions, Sarah. I mean, what's a clinical question that you would receive?
Okay, so if we were to go back to the lymphoma ones, so the patient presents with decreasing vision and has so-called floaters, hasn't responded to steroid therapy because might have been a question of it being an inflammation, so they're given the patient steroids. There's been no response, so then the next thing is, could it be lymphoma? Okay, so that's all written on the request form, and it could be that this is the first time that the patient's had lymphoma, or it could be that 5 years ago the patient had a systemic lymphoma, so a lymphoma elsewhere in the body, and is this lymphoma spreading to the eye? So, on that request form, hopefully there is some clinical information which provides the background for us. Then, when we receive the sample, we say, okay, there's this background, and this is what the clinicians are thinking. They're thinking that this is what they call differential diagnoses. They're thinking it could be lymphoma, or it could be inflammation, which is just not responding. Okay. In the case of the uveal tract, so you might have at the back of the eye, you have the choroid, and you might have a lesion, a lump in there, and the clinicians, the ocular oncologists, they're thinking, well, this could be a melanoma, or it could be, perhaps the patient had history 5 years ago of breast cancer. Okay, and the patient was treated with breast cancer, but could this be a recurrence of the breast cancer in the eye, which can happen as well. So,
wow. I don't want to terrify everybody.
Sorry. Yeah. So the ocular oncologists go in, they do a biopsy, and they say likely melanoma, but previous history of breast cancer. Okay. So so the sample comes in the pot with the details, goes to the laboratory. There are biomedical scientists in the laboratory. In our specialist ophthalmic pathology laboratories, we usually have a specialist biomedical scientist. We look at the specimen together, and what we do in Liverpool is that we create from this tiny little intraocular biopsy, which is like a fluid, we create what's called a cytospin. We spin it onto a glass slide, and this glass slide is thin enough for me to look at it through my microscope. So you can see my microscope behind me there. And so a light source comes up through the glass slide, and I can look down through the oculars, and I can look and see what those cells are on that slide. Okay, so it's prepared for.
And immediately, by looking at those cells, your knowledge can determine, can it? What it looks like. So that's a melanoma, that's a lymphoma, that's metastatic breast cancer in the eye. Who knew?
Yeah. So I guess when you work for probably greater than 25 years now in ophthalmic pathology, it is like pattern recognition. It's a bit like, I don't know how many of these biopsies I've seen in the meantime, probably 3, 4, 5,000, and they obviously vary considerably, but you do recognize the shape of the cells, and also the color of the cells. Melanoma, they have melanin granules in their cytoplasm, and this gives them a sort of slightly darker coloration on the slide. And you can, for the most part, differentiate between a melanoma versus a lymphoma versus a metastatic carcinoma, let's say.
And from that, just concentrating on the uveal melanoma or ocular melanomas, people hear the terminology BAP1, they hear the terminology chromosome 3, it's got 8qa gain. Can you explain to our listeners who kind of know nothing about the makeup of a tumour, kind of how much goes into a uveal melanoma being a uveal melanoma, and actually what that means for us? So however you'd like to start.
So I guess what I was just describing was, when they take the intraocular biopsy, then I'm confirming the diagnosis. Okay. So that's the first step. But then, as we all know, we're trying to squeeze more and more and more information out of these biopsies, whether they're the tiny intraocular ones or whether they're the larger ones. And we're trying to squeeze more information out of that in terms of what everybody calls personalized medicine or precision medicine, because not every tumor is the same. Even though uveal melanomas look similar under the microscope, what is actually going on in the intracellular and subcellular level, that differs between patients. Okay. And so the only way we can really try to discover that is by doing 2 additional studies. And there are different ways you can do additional studies. There's one process called immunohistochemistry. I know it sounds a long word, but it's essentially where we do additional special stains to try and subtype further that uveal melanoma. Okay. With immunohistochemistry nowadays, be it on these small intraocular biopsies of the larger choroidal melanomas, the enucleated eyes, we can do immunohistochemistry for BAP1. Okay. So BAP1 is, I'll keep it relatively simple. So BAP1 is an important gene which is located on chromosome 3. And if it is changed, if it's mutated in a way, then it can lead to the tumor cells being more aggressive. And then when I say more aggressive, that they then are likely to spread within the body and or metastasize. And as probably most people know, there is this something that we can't explain clearly, is this why the choroidal melanoma or uveal melanoma cells spread to the liver. Okay.
Do we not know that? I thought we had an idea that it was that I've always said, oh, it's the next port of call in like there's a tube in my makeup artist naivety.
No, I mean, we don't 100% understand why it goes there. Okay.
Gosh. Okay.
So it goes into the bloodstream, but in the bloodstream, it could spread anywhere. Okay. The tumor cells, but it does seem to have this love for the liver, but there are a number of reasons why we think it does like the liver environment. Okay. But okay, so getting back to what we do with the samples, so we can do either the immunohistochemistry and just look at BAP1, and it's the staining pattern that we can observe within the cells, which helps us determine whether the BAP1 gene is intact or mutated. Okay. There is also the molecular testing, and this is where we get onto the chromosome 3 and the other genes. Okay. So in the 1990s, using a less sophisticated but still very valuable method, a group in Germany, a group in Sheffield, and a group in Vancouver almost simultaneously demonstrated that if you have an alteration of chromosome 3 in the tumor cells only, not in the patient's normal blood cells or in, if you have an alteration in the tumor cells with the loss of one copy of chromosome 3, which is called monosomy 3, then these patients have a higher risk of metastasis. Okay, so this was demonstrated before BAP1. This was demonstrated really in the 1990s. And as a result of this, Bertel D'Amato was the first one worldwide to organise with the Royal Liverpool University Hospital at that time to get consent from patients who'd had their eye enucleated to do analyses for chromosome 3 status. But then over time, again, these researchers in the background were also demonstrating it's not only chromosome 3. Chromosome 1 was important, chromosome 6 was important, and chromosome 8 was important. These tests started, expanding and not only looking at chromosome 3, but also looked at particularly chromosome 6 and chromosome 8. Okay. And so if you had a uveal melanoma which had a loss of chromosome 3, monosomy 3, but you had weird changes in the tumor cells where you had additional copies of chromosome 8, then that combination of chromosomal alterations within the tumor cells more often than not, unfortunately, led to the patients developing metastases. Okay. So it was a way of stratifying patients into good and not good prognostic groups. Okay. And
which back in the 90s, I mean, bless them, there was absolutely nothing we could do about it if it did spread to the liver anyway.
Yeah. But the thing was, people said, well, what's the point in doing this?
Yeah.
But that was very sort of fatalistic viewpoint, because as we know in other cancers, so for example, breast cancer, they have been able to develop magic bullets by discovering certain things on the tumor cells, like the so-called HER2 receptor. And you can now target that HER2 receptor using particular drugs. And, it has really changed around the prognosis for those patients. So we just, if you continue sort of understanding the biology, the underlying biology of the tumor cells and how they differ, that you have disomy 3 uveal melanoma cells.
Hang on. Where did disomy 3 come from?
Sorry. Okay. So I should just go back again. So monosomy 3 is where you have a loss of one copy of chromosome 3. If you have 2 copies, which are normal in the tumor cells, that's called disomy 3. Okay. Right. Okay. So if you have a uveal melanoma, which is disomy 3, no alterations in chromosome 8, but it might have changes in chromosome 6, which seem to be protective, then that tumor is less likely to spread. Okay. So towards the end of the 1990s, early 2000s, you had a very simple sort of stratifying, algorithm, if you want to use that term, where you'd, divide the patients into different groups. And if there was a low risk of developing metastases, then those patients would have ultrasounds of their livers, and it would perhaps start with 6 monthly, but then it'd go to yearly, and then after a certain time, it may stop altogether, because the risk of them developing metastases was really, really quite low. And as you can imagine, the anxiety every time you come back and have an ultrasound of your liver is really can be quite overwhelming, and for some patients.
Yeah, I was going to say, I mean, the thing about knowing that even when there wasn't actually something that could be done about it back then, at least you could continue your life. You could plan, you could go on, you'd have children, you get married. You're not forever thinking.
Yeah, exactly.
So,
you're not left in limbo.
No.
Whereas, patients who have tumors which have monosomy 3 or monosomy 3 plus changes in chromosome 8, okay, they then know that, yes, they do have a bad tumour, a bad uveal melanoma, but therefore they can plan. And I think what Bertel introduced into his clinic when they started doing this, and also I should say, such so-called high-risk patients would then have more regular screening using MRI with the aim that the patients could be recruited to clinical trials or be referred to the liver surgeons and undergo liver surgery as soon as anything came up, whatever size. Okay. And so both Liverpool and Paris demonstrated that there was prolonged survival of the patients if those metastases were picked up earlier and the liver surgeons went in and resected some of those metastases earlier. And it also gave the patients where the liver metastases weren't resectable, they could be, if they wanted to, be registered within a clinical trial. Okay.
When did clinical trials start? I mean, we're still back in the 90s here, so the clinical trials for uveal melanoma have been going 25 plus years.
Yes, and so
wow.
I think the thing is that uveal melanoma has always suffered a wee bit in being sort of like an overlooked cousin of skin melanoma, and then the clinical trials at the beginning just in a way applied the same medications to the uveal melanoma patients that they applied in the skin melanoma patients. And it wasn't until, again, researchers understood the biology that the skin melanomas and the uveal melanomas are completely different diseases. And that, uveal melanomas hardly ever have a certain alteration called BRAF, which is the mainstay for a lot of the treatments of metastatic skin melanoma, and just doesn't work with metastatic uveal melanoma. And so even the 1990s, early 2000s, even probably 2010 or so, until biology researchers demonstrated this initiating genes that led to the development of uveal melanoma, this logic of the application of clinical trials was, okay, let's just try whatever we're using in skin melanomas. Let's just try them in metastatic uveal melanoma. And it was clear over time that it just didn't work. It didn't work. And it's because we didn't understand the underlying biology. But now, really, over the last 10 to 15 years, there's been so much work into understanding the underlying biology of uveal melanoma, the initiating mutations, the additional mutations, and as a result of that, then how we can attack the tumor cells using what mechanisms, which are completely different to the mechanisms which are used in skin melanoma. So, so sorry, I'm going to go back to the...
Yeah, no, I was going to bring you back. So we've done our chromosome 3, 6, and 8.
8, yes. And then...
we left BAP1 on the shelf.
Yes, we left BAP1 on the shelf. So BAP1, the immunohistochemistry which I was describing, is not deep diving into the genes and doing molecular testing for that. So uveal melanoma, yes, is unusual in that it has only about 7, 8 genes which are mutated or changed. And to really understand which ones were mutated, it did actually require an international collaborative study. And that was the Cancer Genome Atlas, which was run in the States, run by the National Institute of Health. And it was the TCGA examined 33 different tumors, and the 33rd tumor was uveal melanoma. Okay, just because they just had a bit of leftover money which they needed to use, and so 80 uveal melanomas were examined, They were very strict in collating that material. For every single 80 tumours, that you had to have high quality tumour material. You had to have you had to know everything about the patient. Anyway, so we in Liverpool, our Ocular Oncology Biobank contributed 25 of those 80 tumours, and essentially all of those 80 samples were studied top to toe, looking from what we would call protein level, going deep dive, really deep dive into the subcellular content of each of the tumor cells, and it came up with and demonstrated, yes, those particular chromosomes were important, so chromosome 1, 3, 6, and 8, but very important was BAP1, another gene called SF3B1, and another gene called IFAX. Okay, so they're the main three ones, but there are other rarer important ones as well. And so the TCGA essentially then divided the uveal melanomas into 4 subgroups. Okay, and so TCGA studied use very sort of sophisticated tools, which were quite expensive and things that you can't apply in everyday healthcare. That being said, like everything, like advances in your phones, your mobile phones, your televisions or whatever, the technologies which are used for molecular testing has also, has increased and become cheaper over time. So now there is something which is called next generation sequencing, which is abbreviated as NGS. Okay. You have next generation sequencing panels which are designed for particular cancers. They're bespoke for particular cancers. So in the meantime, people in the States, people in the UK, people in Europe have designed next generation sequencing panels which look at the chromosomes, and it depends on the panel, but let's say 4 or 5 of those key genes in uveal melanoma, and look to see whether they're mutated or not. And on the basis of this more detailed information, we can better stratify the uveal melanoma patients, those with high risk and low risk of developing metastases. And on the basis of that, then do the surveillance screening.
So next generation sequencing is now regularly used. That's what happens now, is it?
So in the UK, in what is called, they're called genomic laboratory hubs, sponsored by the NHS, there is a so-called test directory where it's required to ensure that there's equity across the UK in what's being offered to uveal melanoma patients, that these chromosomes are looked at and also these genes are looked at, and the next generation sequencing panels are probably the most accurate ones.
And with the results from that sequencing being so personalized, which is where we're going with medicine, are we at a point yet where there is something that can be offered to so say you've had an intraocular biopsy done, you've discovered that yes, it's definitely melanoma, it's definitely a malignancy of the eye, you've broken it down with next generation sequencing. Are we still heading down the pathway of enucleation, radiotherapy, proton beam, brachytherapy? Are we still going down there, or are biologicals catching up with us?
So at the moment, we don't have, none of these genes are ones which are targetable using treatment. Yeah. So for example,
it's just not fair.
Yeah, I know. Yeah. So with the skin melanomas, just going back as an example, so the BRAF gene, which is mutated in about half of the skin melanomas, that is a targetable gene. Okay.
And by targetable, you mean there is a drug that goes in and kills it?
Yeah, a drug which goes in and it concentrates in that area of the tumor cell, and yes, ultimately leads to the death of the tumor cell. Okay, we don't have something like that at the moment. However, what people are trying to do is trying to attack the tumor cells in differing ways, and so for that I might have to explain. So we've got the tumor, but surrounding the tumor, now perhaps we'll concentrate on the tumor which has spread to the liver. Okay. So you can imagine that tumor cells, they want to survive. Okay. So, and we have to imagine that they are, and this is the terminology which is used, they are colonizing the liver. Okay. So they're going there, they're wanting to set up their new camps there in the liver. And so they are then creating their own defenses. And how they can create their own defenses is by creating what people call the tumor microenvironment. So the tumor microenvironment can consist of the tumor cells creating fibrotic walls, creating a castle. Okay. So it's gone, it's spread, it's set, the tumor cells are in the liver, but then it creates walls around it so that the immune cells of the body can't attack those tumor cells. They can't get through those walls.
Okay.
And so that the tumor cells secrete substances which then lead to these sort of fibrotic walls that surround them.
Are they visible to you under microscope?
Yeah, they
wow.
are.
Okay.
Yeah. So that's like if you have a scar on your skin or anywhere in the body, that scar consists of fibrosis. And so essentially what the tumor is doing is it goes to the liver and it tries to set up this fibrotic wall around it. And
around
it's
each metastases or each cell?
Each group of cells. So
okay.
And another way that it can sort of dodge the immune system is by secreting substances which almost paralyze the immune system. So when I say the immune system, I mean the white blood cells. So you've got your B cells and you've got your T cells, and more often than not, it's the T cells which respond to any foreign body coming. The tumor cells are essentially like foreign bodies in the liver. And
now, a T cell isn't a tumor cell, is it? It just starts with the same letter. Okay, just clarifying that. Yes.
No, it's the T cell are what we have in our immune system, which fight viruses, fight bacteria, etc. So it's like an automatic response. The tumor cells have landed in the liver. They might have destroyed some of the liver cells. The liver cells are sending out signals saying, help, 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 something's happening. And then you have this influx of your immune cells, which include predominantly your T cells. And they're trying to get rid of those tumor cells.
I'm picturing them all on horseback with little bows and arrows trying to get in.
Exactly. They're like soldiers. They're coming, they're trying to get rid of those. It's a very good analogy. Trying to get rid of those tumor cells with their arrows. Okay, but the tumor cells are either putting up shields in terms of the fibrotic walls, or they're shooting arrows back, but they're paralyzing the T cells. The T cells become exhausted. And so you hear this expression that the immune cells, the immune population has become exhausted and they can no longer function to get rid of those tumor cells. So this leads me to how, some of the companies are trying to fight the metastatic uveal melanoma cells is by reactivating those T cells, sort of coming in behind them with substances, giving them, I don't know.
He's bringing the drones in.
Yeah, exactly. Sort of bringing them with nutrition, with, gels to keep them going on with the marathon, whatever. And so this is the principle behind, with tebentafusp. It comes and it's latching onto various receptors on the T cells, and it's trying to reactivate them so that they recognize that those melanoma cells are foreign, and together they're combining to destroy the melanoma cells. Okay.
Wow, I had it completely the wrong way around in my head. I thought something like tebentafusp went in and did something to the tumour so that the T cells could then fight. But actually, it's the T cells that it's working on.
Yeah, it's a combination of the two. So it's
okay.
bringing the T cell and the melanoma cell together, and so you've got sort of two little receptors working simultaneously. And the similar principle with PRAME. Okay, so I know we haven't mentioned PRAME here yet, but it's a similar mechanism with PRAME. Okay, so that's one way people are thinking of how you can defend the body against the melanoma cells and get rid of the melanoma cells.
Quick question. That metastatic lesion that is in your liver, will that contain, if you've had in your eye BAP1, monosomy 3, chromosome 8, will your metastatic lesion be the same molecular genetic makeup as your primary lesion?
Yeah, so that's really interesting because for the most part in metastatic uveal melanoma, the changes you see in the primary tumor are pretty much the same as what you see in the metastases. And so that's why it's really important. We'll come on to this probably with the biobanking. It's very difficult to get so-called matched pairs and even have the bloods of the patients. So you can also look at what's going on in the blood. But
difficult for who and in what way? For you in research?
In research and obtaining that tissue. Okay. But the studies that we've undertaken and others have undertaken have demonstrated for the most part that it's almost uniform what you see in the primary and the metastasis. However, the metastasis does continue to evolve, and you might get additional changes. So additional changes, for example, in chromosome 8, that is the most common, that you get more and more copies of chromosome 8. Whereas in other tumors, let's say, for example, kidney cancers and lung cancers, this has been demonstrated by other researchers, one particular, Charlie Swanton, who has demonstrated that over time that metastases can continue to evolve. And in those other cancers, it's very much dependent on where they've metastasized to and to what organ they've metastasized. So, but uveal melanoma is relatively stable
and quite simple. I've heard it's classed as a low shedding tumor.
Yeah, that's right. It has a low tumor burden, and in terms of low tumor mutational burden, because there aren't many genes which are mutated.
Is that better for the patient cohort than having a complex tumor, or does that actually limit our options of treatment? Sorry, that was a complete curveball question.
Yeah, no, it does limit in a way, because if you have, say, for example, a colorectal carcinoma or a lung carcinoma where you've got mutations which are targetable, then you can go in and you can hit one gene, and if that doesn't work, then you hit another gene, and then you. So whereas with uveal melanoma, yes, there are unfortunately not so many mutations present, and of those mutations present, there at the moment they're not targeted. Okay. So there are, however, medications on the horizon which are using what are called attacking the signaling going on within the cell. And this is darafusertib, which is very difficult to pronounce, which is inhibiting certain molecules within the tumor cells. It's called a PKC inhibitor. It is being combined with another medication, which are just for simplicity called Daracris, and that's being used in the metastatic scenario. But there are promising results, both with, as I mentioned, tebentafusp. PRAME is still being tested, so to say, in clinical trials.
Yeah, what is it? What is PRAME? Is that an acronym?
Yes. PRAME is an acronym which stands for Preferentially Expressed Antigen in Melanoma. And it helps, again, with a stratification of the patients into low and high risk groups in terms of onset of metastasis. And the PRAME, a bit like the BAP1 immunohistochemistry, we can do PRAME immunohistochemistry on
just on the primary.
On the primary, yeah.
Okay,
on the primary.
yeah.
Although when patients develop metastases in the liver, you can do a biopsy of that liver metastasis, confirm again that it's melanoma, because some spots on the liver could be something completely different. And you can do the BAP1 immunohistochemistry, and you can also do the PRAME immunohistochemistry on both of those. Okay, so you can examine PRAME using immunohistochemistry, but you can also look at it at the microRNA level, and that's being undertaken more often in the States. So the mRNA expression is being examined using kits which have been developed by Castle Biosciences, for example. Okay, but for the most part, there is reasonable concordance between the immunohistochemistry for PRAME with the mRNA expression. Okay, and based on that, then the patients, as I mentioned before, are stratified either into low or high risk, and there are these clinical trials for patients who have PRAME expression in their melanoma cells that could be entered into these trials. Okay.
Wow. Okay. So the future is getting brighter. That's one thing for certain.
Well, I guess when patients may have heard of this term biomarkers. So what we're trying to do when we're diagnosing, doing all these different immunohistochemical and molecular studies, is we're trying to identify biomarkers, which could be then used for A, stratifying the patients into risk of metastasis, or B, stratifying them in terms of whether they are suitable for treatment with certain agents, therapeutic agents. And both with the PRAME and the tebentafusp, there is a biomarker in the blood which has to be also taken and measured, and that's a certain HLA, what's called a haplotype in the patient's blood. And if that is present, then that patient would be eligible to go into the trials, looking at tebentafusp or using the PRAME therapy. Okay, I realize I'm getting into sort of complicated areas now, but
that's okay. I mean, the HLA type, I think I've spoken about that in previous podcasts, but for those who haven't listened to those before, this is done via a blood test. We're not talking biopsies to determine the HLA type, and it's roughly 50-50 of the population has it as a positive. I think it's a protein type, isn't it, within the blood?
Yes.
And the other 50% doesn't. So we're there is a treatment now for the HLA positive in tebentafusp, and things are looking a lot more positive for the negative set of us as well.
Yeah.
So your little biobank.
Yes. So
tell us.
So we must give full credit again to Umberto D'Amato, who started he realized that, the only way we're going to understand the biology of the disease is to consent the patients, not only for the surgery, obviously, and the treatment, but also for harvesting tumor tissue from, for example, from an enucleated eye from consented patients. And so when I first arrived, it was quite small, this biobank, And then I was able to recruit Dr. Helen Callery, who used to work in another biobank within the university. But she has incredible knowledge about all of the rules and regulations that you need when you're setting up such a human tissue biobank. So we have a consent form which has gone through the ethics committee in the hospital. It is based, the actual biobank is based within the university. It is sponsored by both the university and the hospital. And this consent form is usually presented to the patient by the ocular oncology nurses who have been trained by Helen in how to take consent from the patients. Essentially what happens is that, on the consent form, the patient has a list of tick boxes whereby they can indicate what they would like to consent their tissue to be used for. So it could be just within Liverpool, or it could be just within the UK, or it could be also that these tissues could be shared with collaborative researchers overseas and or with industry. And there is also a specific section saying whether the tissue could be used in animal studies or not. And, so all of the patients have the options to indicate to what extent their tissue could be used. So we have a setup here in this building where I'm sitting in the university. So we have freezers which contain all of the deep frozen tumor tissue, and every patient's tumor tissue is clearly marked. And more often not, accompanying that tumor tissue, that frozen tissue, is a blood sample. And through the consent form, we also have access to the paraffin blocks. So I might have, at the very beginning, I mentioned that tumor samples or uveal melanoma samples can be sent in a solution called formalin, which is a preservative. That sample then comes into the laboratory. The specimen is then embedded in wax, in cassette, and from those it's from that embedded tissue that very thin slices are made so that I can look at the slices of the tissue underneath the microscope. So they're 4 microns thick, so they're very thin and allowing light to come up through. Okay. So I use those for a enucleated eye. I use those paraffin blocks and those microscope slides to make the diagnosis, to do the immunohistochemistry, to do the molecular studies. Okay, but when that's all finished, and if the patient has consented for the use of the tissue for research, then I not only have access to any frozen tissue, but I also have access to all of those paraffin blocks for further research downstream. And so in our ocular oncology biobank, we have large numbers. Estimate is about 3,000 worth of harvested frozen down tissue, tumor tissue, accompanying blood samples, and also paraffin blocks. Okay.
Do you ever get the matching metastatic lesion blocks sent up to you as well?
Yes. So our consent form does enable us. So should a patient develop metastases to the liver, those tissue blocks are contained here within Liverpool. Then because of that consent, we have access to those paraffin blocks of the liver metastases.
So that's only if the metastases has been dealt with in Liverpool, though. You can't, people can't post their bits to you.
We can organize if the patient has consented and say, for example, they're in Birmingham and they develop the metastases, then because we have the consent, we can organize what's called a material transfer agreement between the hospital in Birmingham and ourselves, so that we can access that metastatic tumor tissue in the liver, and so that we can then coordinate any research on those specimens.
Is that useful for you, Sarah, to have
Definitely.
that?
Definitely.
Okay.
Yeah.
And how would someone go about arranging that?
So It has happened in the past. So patients who are wanting their metastatic uveal melanoma samples to be used for research, they know that they've consented here, then they can organize, they can contact ourselves, myself or Dr. Calleray. It does take a bit of paperwork, but we can organize the material transfer agreement so that those tumor blocks are sent and are then contained within our ocular oncology biobank.
Okay, well, I think if people want to do that, then I think if they emailed the charity through OMUK,
Yeah,
I'll put the email address up and maybe, I mean, I'm sure there would be people out there who were desperate to actually give you as much information as we can, and if that includes bits and bobs that are in laboratories on shelves elsewhere in the country, it makes an enormous sense to have things centralized with you. Sorry, I continue. Please go on.
No. So I think this ocular oncology biobank now is close to 15, 16 years old. We've been able to renew its ethical approval, and this happened very recently, so it's been renewed for another 5 years. And to be honest, there's nothing similar in the UK, and I think the closest similarity would be in Paris, where I know that they also have a biobank of sorts. What we've done more recently is we've expanded the consent to also store in what's called a trusted research environment the digitized images of all of the histology slides of the tumors, the digitized images of all of the fundoscopies, and also digitized images of the surveillance scans of the liver. And so for every single patient now, we're hoping to almost have 4 or 5 dimensions so that we have all of the clinical data, the basic data in terms of age, gender, the usual data that we have, the genetic data in terms of, the chromosomal changes and the mutational data. Then we'd have the digitized images, the histology, the ocular images, including the OCTs and also the MRIs of the ultrasounds. And so this is what we've called ICAN-AID, so eye cancer artificial intelligence digital bioresource, or ICAN-AID for short. And it is a trusted research environment, which the governance of which is very similar to the Ocular Oncology Biobank, sponsored by both the university and the hospital. There would be, if researchers would like to, for example, just examine the ocular images. We have a PhD student, how can we differentiate between a small melanoma and a nevus? How can we apply new AI techniques to help, differentiate that? We have Max Jackson. He's done a similar study and actually published it as well.
Okay.
Where you are applying, different algorithms, some developed by Moorfields, Piers Keane, and his group, seeing if you can pick up or even differentiate, a metastatic carcinoma to the back of the eye versus a melanoma. So all of these different things. Or, somebody wants to just look at the digitized histology images or the digitized cytology images, all of that can be accessed if people would like to, or researchers would like to have access to, I don't know, let's say 1,000 images. It goes to the governance committee of the ICAN-AID, and if there's a good track record and the project makes sense, etc., then those images access is provided for that person to look at those pseudonymized images to undertake whatever research they're wanting to do. So this is essentially an expansion of our ocular oncology biobank. It's not only looking at the biology, but it's also looking at how we can apply new techniques to better understand the tumor cells, but also improve diagnostics, earlier diagnostics.
I mean, it's a kind of stupid question, really, but why is this so important in a rare cancer?
I think just because you've got a rare cancer doesn't mean that you shouldn't be treated as equally and seriously as if you're a patient with a common cancer. And if you were to put all of the rare cancers together, I think I can't remember the actual statistics, but I think it's like 25% of, if you combine all of the different rare cancers and rare diseases together, they, affect at least 25% of the population. So I just sort of feel that, that you shouldn't be overlooked or undertreated because you've got a rare disease. What is really important with these biobanks, and particularly with the rare cancer biobanks, is that it's only through such collections and collaborations with differing research groups that we're going to be able to make any breakthroughs, that we're going to be able to come up with magic bullets which are required to cure metastatic uveal melanoma. And, I think the infrastructure in England with these 4 specifically funded ophthalmic pathology centres, together with the 3 ocular oncology centres, we should be really making best use of that to set up such biobanks and ultimately connect them. I mean, my dream would be that we have a national trusted research environment for ocular melanoma, and where we'd have digital bioresource of all of the patients with ocular melanoma, uveal melanoma, where possible, going forwards, we, do all of the molecular genetics. We understand, we follow them also longitudinally and have the survival data. We create such what I would consider a gold mine so that, researchers can examine different aspects of uveal melanoma and try and crack the code so that we can come up with treatments to improve patient outcome.
I mean, that is a wonderful thing to work towards, and I hope that happens. I mean, so if somebody has their treatment in Moorfields and has their eye enucleated, would they be offered the same consent, Sarah, so that their tissue can be donated to the biobank once it's undergone its testing and everything?
Moorfields does have a biobank in the meantime. So the Liverpool Ocular Oncology biobank consent form couldn't be used in Moorfields, but I think, and I have to double check this, but I think they have a similar consent form, which mirrors very much what we have in Liverpool.
But is there a collaboration between the two of you? Because if you've got 3,000 and they've got 3,000, I mean, the population down here is like 9 million. I know people come in from Europe as well. Then stronger together is better.
Definitely. In Moorfields, there wasn't sort of a consequential collection of ocular melanoma in the past, but over the last, say, 3 to 5 years, they have been collecting ocular melanoma tissue for research. And as I mentioned before, you can set up material transfer agreements between hospital trusts, so such that there would be, we could organise transfer of material from Moorfields to Liverpool or vice versa, for research. So
I've just got this image of like a little bike with a back box on it and all these little eyeballs just sitting in it, heading up the M1. But I'm sure that's not how it works at all.
No, yeah.
But I mean, collaboration surely is key in this. And are you working internationally as well? Does the ICANAID and the Liverpool Biobank, are you part of an international conglomeration of body parts?
Yeah. So before the pandemic, our research group was very much involved in what was called Horizon 2020 European project, and that involved 12 organisations across the EU. And it involved Manchester and Liverpool from the UK and 10 other centres across Europe. And that was a 5-year study, and there was approximately 6 to 7 million worth of euros, because it was sponsored by the EU, into various aspects involving research in uveal melanoma. It was interrupted, unfortunately, during the pandemic. However, it did come to a conclusion in 2021, I think. So that's just one example of how we collaborate. And we've collaborated also, I mentioned the TCGA, but we do collaborate with other researchers in the States, actually also in Australia. I'm just sort of thinking Singapore. We are conscious of the fact that we don't have all of the technologies here in Liverpool to do all of the different studies. And so it makes total sense that we share the samples with these other centres who have, either have, for example, licenses to animal studies, or they have more sophisticated technologies than we do, and to sort of delve into certain areas of uveal melanoma. So we're more than willing to share the ocular oncology biosamples and also the images, because in the end, we all want the same thing. We want a better survival for the patients and ultimately, ideally, better treatments for patients with primary and metastatic uveal melanoma.
And I suppose that would be your closing message to people newly diagnosed today listening to this. It's not as bleak as it was, most definitely,
Yes.
with research now.
No, I think I'm always optimistic with things. I'm a person with a cup which is half full. And I know that's difficult for patients, people who have cancer, to think positively, but I can only try to reassure them that there are many people who are beavering away and trying to, progress things, even if it's just millimeter by millimeter. And sometimes you have a sudden jump and it's a whole meter and you have a breakthrough. But 30 years ago, skin melanoma was considered to be incurable, and other, cancer types, for example, Hodgkin lymphoma, were considered to be incurable. In the meantime, we know that you can cure people with both of these diseases, and I'm sure it will be the case with metastatic uveal melanoma as well. We just got to not give up hope, got to continue in believing that there will be a cure. And that's what I guess I've been working towards over the last 21 years. So.
Well, we thank you so much for your service, your time, your input, your effort. And I would imagine many a sleepless night and long-standing shift in a laboratory staring down the eye of a microscope. Professor Coupland, Sarah, thank you so much for joining me today on iConnect.
Oh, well, thank you very much, Joanne. It's been an absolute pleasure. Thank you.
If you or someone you know has been affected by the topics discussed in this episode, support and information are available. You can visit Ocular Melanoma UK at www.omuk.org for guidance, resources to connect with our helpline, and others who understand. Remember, no one should face ocular melanoma alone.